Thank you and welcome. My name is Janet Marchibroda. I'm the president of the Alliance for Cell Therapy Now. We're an independent nonprofit coalition of primarily academic and research institutions who are working to advance the use of regenerative medicine and cell therapies for patients in need. We're so delighted to be here at the World Stem Cell Summit and the Wake Forest Institute of Regenerative Medicine, their Essentials course. And we're especially delighted to have this esteemed panel of leaders from the National Institutes of Health who will share their insights with us during our panel discussion, which is entitled Exploring the Use of Regenerative Medicine and Cell Therapies for Serious and Life-Threatening Conditions at the NIH, the National Institutes of Health. So really quickly, joining us today for this panel discussion is Dr. Ilyas Singec, Director of the Division of Preclinical Innovation, Stem Cell Translation Laboratory at NCAT, the National Center for Advancing Translational Sciences at the NIH. Also joining us is Dr. Lillian Shum, Director of the Division of Extramural Research at the National Institute of Dental and Craniofacial Research at the NIH. And then also so delighted to have Dr. David Stronzek, who's the chief of the cell processing section at the NIH's Clinical Center. To kick things off, I'm hoping that each of you can tell us a little bit about yourselves and your role at NIH and what you're doing. And I'm hoping, Dr. Singec, that we could start with you.
Excellent. Yeah, thank you very much, first of all, for having me. And it's really a great honor to be on this panel and have this conversation with you. So I'm part of the Division of Preclinical Innovation. However, I'm not leading that division. So I'm actually, my group, the Stem Cell Translation Laboratory, is part of the Division of Preclinical Innovation at NCAT's NIH. So a little bit about my background. So I have a neuroscience and neuropathology background. This is how I got interested in stem cell biology. So as a physician scientist, I was always motivated to work on clinically relevant questions and problems. That's how I really decided many years ago to stay in stem cell research. And so I joined NCAT almost 6 years ago now to start a brand new laboratory that we call the Stem Cell Translation Laboratory. So this was supported by NIH Common Fund, and it was really intended from the very beginning to help to translate induced pluripotent stem cell technology into clinical applications. So we're doing this in a collaborative fashion and trying to address the key technical and scientific obstacles that so far have been challenging for the entire field. So areas of interest are introducing quality control standards, for instance, developing improved cell differentiation protocols, deep cell characterization using novel technologies such as single cell analysis. So also to develop new drugs that we can use to better control stem cell behavior. So additional areas of interest are what we call ad hoc collaborations. So we have ongoing efforts in Zika virus biology and also looking at repurposing drugs for the current coronavirus pandemic. And then lastly, we also became part of the HEAL initiative. So HEAL is a new, relatively new initiative at NIH. HEAL stands for Helping to End Addiction Long-Term. So this is a response to combat the opioid crisis that is, as you know, devastating in this country. So this is, yeah, kind of a brief overview of what we are doing at my group at NCAT.
Thank you, Dr. Singec. Dr. Shum, could you share a little bit about your work and about yourself for our audience?
Thank you. Thank you for having me. It's a real pleasure to join this panel. As Janet mentioned, I'm the director for the Division of Extramural Research at the National Institute of Dental and Craniofacial Research, or NIDCR, one of the many institutes at NIH. My job is to provide leadership and direction for NIDCR to invest in a broad spectrum of basic translational and clinical research related to dental, oral, and craniofacial health and disease. The division has 16 programs ranging from microbiology, immunology, genetics, to data science, behavioral science, clinical trials, health disparities, and so on. Obviously, we have a program in tissue engineering and regenerative medicine that is of primary interest to this audience. And in addition to supporting research and research training, we also develop targeted initiatives to accelerate research in high priority areas, to fill knowledge gaps or capture emerging opportunities. So for example, this year, we launched an initiative to support research on defining lineage plasticity and endogenous regeneration capacity.
Thank you, Dr. Shum. Dr. Stronzek, could you share a little bit about yourself and your work at NIH?
Yeah, I lead the cell processing lab. I lead, we process hematopoietic stem cells for transplantation at the NIH Clinical Center. We also do manufacture a lot of cells for early phase clinical trials. We're involved in phase 1, phase 2 clinical trials for patients enrolled in protocols at the NIH. We don't manufacture cells for patients at other institutions, and we wouldn't do any phase 3 or 4 manufacturing for phase 3 clinical trials. The types of cells we manufacture is quite variable. We do manufacture a lot of cancer immunotherapies, a lot of CAR T cells and TCR engineered T cells. In addition, we do some genetic engineering of hematopoietic stem cells to treat children with immune deficiencies, but we've also been doing some regenerative medicine studies. In the past, we've manufactured bone marrow stroma cells to treat patients with graft versus host disease or ischemic cardiac disease. Now we're in the process of manufacturing induced pluripotent stem cells and differentiating them into retinal pigmented epithelial cells to treat patients enrolled in protocols in National Eye Institute to treat patients with age-related macular degeneration. So, personally, my background, I am trained in hematology and oncology. Got interested in transfusion medicine, so did a little cell therapies at the University of Minnesota and some immunohematology. For the past 15 years, I've been running the cell processing lab here at the NIH Clinical Center, and the field's been interesting to see the field's change remarkably over that time.
Oh my goodness, so much interest. It's great to hear this. So much interest in sort of optimizing and scaling manufacturing in this area. I think that's one of our key challenges and opportunities. And I know many are interested in seeing your work and the work of your colleagues at NIH. I thought I'd drill down. Dr. Shum, I know many of our participants, looked in great interest. We were flipping through the president's budget, and there was a lot of discussion in the NIDCR budget around how you all have used regenerative medicine and cell-based therapies. Could you talk a little bit, in greater detail about the exciting work that you're doing? And then maybe, what led your center to expand its research in this area?
Sure, thank you. So at NIDCR, our tissue engineering and regenerative medicine portfolio is approximately $40 million. So maybe small relative to other NIH institutes, but large with respect to the proportion because it's about 10% of our extramural budget. And we support research broadly on reconstruction, repair, and regeneration of craniofacial, oral, and dental tissues. For example, skeletal tissues, including the temporomandibular joint, teeth, periodontal ligament, skin wound repair, oral mucosal healing, as well as salivary gland regeneration. And more specifically, we support research on the biocompatibility of materials and integration into the host tissue environment, considering the immunogenicity and biodegradability. We also support research to promote endogenous capacity to regenerate, as well as multiple tissue constructs or biomimetics. Definitely support in the design of scaffolds, not only to provide structure, but functions to lost tissues like smart materials, and even incorporating drug delivery capacity in these materials. Bioreactors, microfluidics, 3D printing, those are all in our portfolio. And with regard to cell-based therapy, we support research in the isolation, characterization, expansion, and differentiation of stem and progenitor cells. Research on cellular reprogramming and homing, including research on stem cell niches, controlled delivery of cells, genes, biologics, small molecules. But I mean, the overall goal is really to restore and repair tissues, both structurally and functionally. We have a long-standing interest in tissue regeneration. And a few years back, talking about expansion and what we invest in, we conducted a portfolio analysis, and that revealed both the need and the opportunity to adjust our investment. So, number one, back then, many of our projects were focused on single tissue regeneration. So we are now supporting more on multiple tissue complex constructs. So for example, not just bone, but vascularized and innervated bone, or not just acinar cells in salivary glands, but whole salivary gland units. Number two, back then, many of our projects were in the discovery space, experimenting with different kinds of scaffolds, maybe changing the materials, the pore sizes, and other physical characteristics. And now we have shifted our investment to more translational aspects, including the use of large animal models. Number three, back then also, many of our projects utilize animal models that do not accurately reflect the human conditions. So for example, regeneration studies in young and healthy animals. And we know that in humans, many of tissue regeneration is needed in less than ideal host environments. For example, in older adults or in environments with comorbid conditions. So studies in more challenging models are needed. So with that portfolio analysis, one specific action that is driving the expansion of research in these new directions is the creation of the Dental, Oral, and Craniofacial Tissue Consortium. The goal of the consortium, which includes 2 resource centers and multiple interdisciplinary translational projects, is to support preclinical IND and IDE enabling studies for regenerative medicine products to get through the FDA so that products can enter clinical trials and first in human use. We're in our 6th year of supporting the consortium, and we are seeing products moving through the valley of death, attracting industry support, and have had productive interactions with the FDA.
That's great to hear. And, we're so excited about your center's focus in this area. And we're hopeful that some of the other centers at the NIH might follow suit. And it's helpful to understand the background. So thank you. Thank you. Dr. Singec, I'm hoping you talked a little bit about the work that you're doing in the translation laboratory at NCAT. And I wonder if you might delve more deeply into the work that you all are doing specifically in this area?
Mhm. Yes. So as already mentioned, so we really focused lab on iPS cell translation. And so the original goals are really around improving reproducibility, for instance, as one main area. So establishing quality control standards, bringing more rigor to the technologies, but also really focusing on innovative technologies themselves. So including finding new drugs that we can use to better control stem cell growth and differentiation. So one example is actually a recent publication that we just put out. So we developed a small molecule cocktail that we named SET cocktail. So this cocktail dramatically improves stem cell growth and differentiation, including cryopreservation and single cell cloning, gene editing, and other applications that we have been improving by using this cocktail. So we think this could be really a platform technology that many in the field could be using. So this is one example, but continuing to improve the overall process of cell differentiation, but also cell maturation are other areas of interest that we are currently working on. So the basic idea here is really to help in a collaborative way, generate data sets that we will publish, but we will also provide all the raw data, for instance, and also provide the foundational data sets and reference data sets. That's, I think, an important missing piece in the field. So ultimately, all this data will be deposited on a new website we're working on. It's called the iPSC Portal. So on this iPSC Portal, there will be tutorials, there will be detailed protocols that can be downloaded, there can be data sets for further data mining, hypothesis building, etc. So altogether, we're trying to leverage areas that have been really difficult so far to be addressed by other groups. And so we're hoping that through this effort, so we can really collaboratively bring the iPS technology together into clinical applications.
Do you have is the portal, is it public yet, or do you have a sense of timing?
So we had a soft launch, I would say. So we are really building this up as we speak. So I think in a couple of months there will be an official public announcement. So that we want to make sure that this should be a useful resource for the community.
It's the whole issue, and I know many of you have talked about cell characterization, but in all of our discussions with the NIH, you've really encouraged us to focus in this area. It's tough, but very much needed as part of our path forward. So thank you. Thanks for your leadership. Any insights about history or what has driven you to focus more in this area? Anything that you could share with us about the why, I guess?
Yeah, so it's really the common problems I think we all have been dealing with for many years. The long-standing problems, difficult to solve, and NCATs in particular, by design, is really set up to help us with the translational process. So just a few comments about how the intramural research of NCATs, it's the division of preclinical innovation, is set up. So we have not only stem cell biologists as colleagues and team members, particularly in my group, but we are also surrounded by chemists, by computational biologists, by toxicologists, by experts in rare disease, therapeutic development. So it's truly a multidisciplinary approach that we're taking, so to make sure that these really big problems are being tackled in an interdisciplinary way, using the best technologies, trying to put forward the best science. And again, everything is around reproducibility, and the other points that I'm sure you will be touching on, particularly the challenges in cell therapy. So I can elaborate more on that.
Dr. Stronzek, could we turn to you? We're watching with great interest the work that the cell processing section at the clinical center is doing to explore things like you talked about development of new therapies, assessing the quality of cell therapies, which we know is a big topic of discussion in the field, and of course, mechanism of action, another big topic. Could you spend a little more time talking about the work that you're doing? Again, sort of how did it come to be, and just what led the NIH to invest in this area?
Well, and the NIH has always had some investment in cell therapies. In fact, the first gene therapy was performed at the NIH in the 1990s. But what's really changed is the discovery of induced pluripotent stem cells about 10 years ago, and then also the success of CAR T cells for treating B-cell malignancies. So consequently, the intramural programs on the Bethesda campus have been really interested in developing both immune therapies for cancer cells and for B-cell malignancies. So they've invested quite heavily in the program. NEI's invested heavily in the program to develop iPS-derived retinal pigmented epithelial cells to treat age-related macular degeneration. So consequently, they've expanded our facilities and increased our staff. We've been interested in markers for potency. So CAR T cells seem to be quite forgiving in their ability to kill cancer. So there can be quite a bit of variability in the manufacturing, yet patients can still get cured. Yet it's important to find out what characteristics of the cells are making them more potent. And similarly, it's even more important for regenerative medicines. It takes us several months to make autologous iPS stem cells. So it's important to find markers that help us predict which clones are going to be the best and which clones to discard. And similarly, when we're differentiating the iPS cells into retinal pigmented epithelial cells, that takes a couple months. So we're looking for markers or measures to see which, we differentiate multiple clones to see which clones will function better. The other thing that's really kind of interested us lately is we've been involved in some collaborative studies. So we would collect cells, send them off to a manufacturer, and then processed cells would come back. And what's the best way to handle these cells? Typically, they're frozen if you can freeze cells, but then each center has a different mechanism for thawing the cells and infusing the cells. And we think there needs to be more standardization of that. So at least every center does it the same way. And then it's even more challenging with some of the regenerative medicines, which methods haven't been developed for cryopreservation. So, do you ship these cells from a central place at room temperature, or do you ship them at 4 degrees, or somehow maybe you can freeze them? And there has to be good methods to ensure that during the shipping process, the cells still retain their potency. So these are some of the things that have been, interesting us. It's kind of nice to see the field's gotten to the point where the production of the cells has really gotten very good. So now we're focused kind of on more uniform manufacturing and more uniform delivery of these cells to the patient.
Could I, because this is of great interest, I know, to a lot of our listeners today, so what is or any of our panelists, what's the path forward when we get to best practices, or you talked about manufacturing, but also the thawing and the delivery process. How does the field move forward in an effective way? Is it sharing best practices? I don't know, a resource center? How do we get there?
I think we've been lucky because I think there's so much money going into regenerative medicine and cancer immunotherapy that companies are stepping up and developing more GM, the methods we're using to freeze and thaw cells were developed in the research lab. So they're developing GMP instruments for freezing cells. We classically manually thaw cells in a water bath. They're developing more mechanisms that don't use water baths to thaw cells that are much more uniform and much more automated. So I think that's one mechanism that in companies that develop devices are making some of these things that help out. I know our investigators are interested in this too in NEI, and they're thinking about ways to ship cells, and the companies that are involved with these phase 3 clinical trials, some of them are actually interested in solving these problems. So, but the way they solve them will probably, it's probably not going to be a trade secret. So they'll probably share that with other groups. So I do think there's a way to this type of the field, it can be more open to sharing and presentation at meetings and other ways to share knowledge. So
that's great. I'm thinking in particular the academic and research institutions who have labs, and whether there's a way to improve. So we've got the folks who are listening today, lots of researchers. They're pursuing phase 1 or phase 2 trials. And if you look at the data in this area in regenerative medicine and cell therapies outside of CAR T, just not a lot of federal funding yet. And so they're anxious. They want to work with you. They love to get support or collaboration. If you're willing, any advice for them when it comes to sort of navigating funding at NIH or even other institutions? This is for all of our panelists, if you're willing to share.
Yeah, I'll give it a stab here. I'm sure many in the audience who have conducted this research are also experienced in seeking NIH funding. So finding funding opportunity announcements or putting together grant applications should really not be an issue. And of course, the NIH website on grants and funding offer numerous resources for every step of navigating through the system, as well as a weekly guide on what new opportunities are out there. Now, I would say that one resource that is underutilized is really us, the NIH staff, especially the program officers managing these clinical trial programs. And we can help researchers to find a good match on funding opportunity announcement, explain to them the Institute priorities, either through our strategic plan or other types of events and campaigns. We can also help explain the requirements of what is expected in a clinical trial application. So, for example, some funding opportunities, they allow a planning stage, while others do not. We can also explain the expectations on specific milestones, whether you need a protocol synopsis, a recruitment and retention plan, certainly a statistical analysis plan, but also a quality management plan or a monitoring plan and so on. And after review, program officers can also help provide feedback on how to interpret reviewers' critiques for you to put together a better application if you need to resubmit. But, in terms of priorities and to get funding outside of, the CAR T area really would require the program office to tell you explicitly what is the institute, what are the institute's priorities.
Thank you so much for that. Grateful for the guidance. Dr. Singet or Dr. Stroncek, anything to add? Any advice for our participants today?
Yeah, it's probably of interest to mention the the Cures Act, so regenerative medicine, so 21st Century Cures Act, regenerative medicine. So their ongoing efforts as part of the it's called the Regenerative Medicine Innovation Project, and also Regenerative Medicine Innovation Catalyst. So this is an effort together with FDA to develop cell products collaboratively, and there are funding opportunities that could be of interest really for the audience. So I would encourage the audience to take a look at these activities as part of the 21st Century Cures Act. So at NCAT, just to mention that, so we have a kind of a different model. So so our intramural research is offering extensive collaborative opportunities. So we could help really at the front end to develop rigorous technologies that would really then be used in phase 1 and phase 2 later on. And this could be very practical challenges, such as making things more cost efficient, for instance, which is obviously a tremendous challenge in the field. So I think a combination of practical thinking, but seeing that these kind of efforts are always long-term commitments. Trying to really engage stakeholders, and there's also hope that maybe at some point there will be more opportunities and funding models, so where public and private sector can come together and support these type of endeavors. So I really look forward to that.
We would love that. Thank you. Dr. Stronzek, any advice?
I'm with the intramural program, so we're not
Yes.
involved with funding of grants. So
Understood. I thought I'd give it a try. Last question before we move to our key takeaways for our audience. Any challenges? Again, our audience, primarily researchers and developers out in the field. From your vantage point, what challenges are you seeing amongst all of us, and any insights about how to address them? Dr. Shum, can we start with you?
Sure. I think, one of the challenges is navigating through the regulatory pathways, especially for those investigators who are primarily in academic research. So, as you know, many regenerative medicine products are combination products. So to get the right designation at FDA, understanding the requirements for GMP studies, safety, not just efficacy, human factors, and so on, might not be familiar territory. So I think it would require reaching out to others, like you said earlier, best practice to be shared, and really shifting the mindset, to product development.
Others, Dr. Stronzek, are you seeing any challenges out there? From where you sit. I know you do some work with private sector.
Well, I think a lot of some important challenges for investigators working in the phase 1, phase 2 space to look ahead. Many of these products are successful and should go on to phase 3 trials, but I think we have to be careful that we use reagents and things that are conducive to licensure and moving on. I think it's important to try and look at systems that keep the cost of manufacturing down. So then they are practical if they are successful. So we've always tried to figure out ways to use closed system processing, some way to try and avoid contamination and maybe keep the facility and quality costs down. So I think these are some of the major challenges for early phase trials.
Dr. Singet?
Yeah, Yeah, just to add to this, and coming back also to our reproducibility challenge in general, I think the entire field still needs to really think about developing the rigor that we have in drug development. So how can we apply those kind of rigorous methods also to cell therapy? I think this is still an ongoing important effort. Another challenge that we have seen is scalability. So making lots of cells in a consistent fashion, and we have some progress using robotic systems. So how can we automate biomanufacturing of cells in a consistent fashion that is investigator independent, and also these protocols have to be adapted first to these kind of large scale approaches. And cost is always a challenge, and not only the operational costs, but also, I would say, the initial thought about what type of cell therapy are we interested in developing? Is it autologous? Is it allogeneic? Is it based on universal cells? And ultimately reimbursement, those kind of general challenges is also something I think that we all have to think about and discuss as a community. But altogether, I would say the last decade has shown tremendous progress in all these areas.
So be patient, keep pushing. These are all things that we need to deal with, and we're just grateful that all of you are helping to sort of pave the way in each of these challenge areas. So we're coming to the end of our panel discussion, and I was hoping that each of you, if we might close with a set of key takeaways for our audience. And Dr. Shum, could I start with you? Any key takeaways for folks who are with us?
Thanks, Janet. I think, mapping out a pathway to success by understanding how similar technologies are approved and adopted would be good. So learning from precedents, learning from best practice, like you said earlier. And certainly number two is tapping into experience in large operations. Like the Wake Forest Institute of Regenerative Medicine would be very productive, or the facilities and consortium that, the panelists here are overseeing would also be good.
Thank you. Dr. Stronzek, any key takeaways?
Well, I think that investigators really have to be patient. We're involved in process and product development, and typically even something that works well in an animal model, it'll take at least a year to get it to develop the scale it up, develop the CMG processes, do the validation studies, and then submit the IND. And then even once the IND is approved, it takes a while to get the studies going and get them done. So and typically for the first few patients will go well, but it's almost always within the first half dozen patients, something unexpected happens. So you almost have to be ready for the unexpected.
Thank you. And Dr. Singet, last word, key takeaways.
Yeah, key takeaways, I would just really emphasize that, patient safety comes always first. And we should really keep this in mind, and not to do harm as the guiding principle. But at the same time, we see that all the tools of the trade are getting better. And so we have been making progress in these various areas of cell therapy, but also other interesting or related areas and technologies such as gene editing is really making great progress. I think combining these technologies and also seeing that, or better understanding the biology and stem cell plasticity, demonstrating more recently that, for instance, iPS cells can give rise to placental cells, so trophoblast. These are very exciting areas. I think we still need to invest more and really also have access to areas that I thought previously we thought it's not possible to generate those type of cells. And this could open up a whole new area of research, such as studying, infertility, pregnancy associated diseases, but also I think continuing to really strengthen collaborations nationally and internationally, and open science, I think, is still going to be very, very important going forward.
Thank you. Thank you. And I'd like to extend a very special thank you to our wonderful panel today, Dr. Singet, Dr. Shum, and Dr. Stroncek. We're so grateful for your time and that you took and the insights that you shared today with our audience. And many thanks to all of you for joining us today at the World Stem Cell Summit for this terrific discussion on exploring the use of regenerative medicine and cell therapies for serious and life-threatening conditions at the National Institutes of Health. So thank you. And this concludes our session. Thank you.